Thanks again. Okay, really appreciate the chance to talk to everybody today. Um, and I'm going to talk about marine pest management and good scheduling there to be after Chris, because one of the key sort of gaps I think we have is what are the range of options we have when we actually detect a pest. So Chris just described. I think we can sort of take it for granted sometimes in New Zealand that the consistency of the funding, the consistency of the effort, all the expertise that goes into it. That's really, really world class delivery of surveillance. And we always have to do work to sort of keep at a world-class level, but that's really sort of impressive. But when we get to a species that is here, it is deemed to be problematic, and we want to start doing something about it, our options start to diminish relatively quickly for marine biosecurity. And so that's some of the work that we're working on at Cawthron in our program. So marine pest management is lagging behind terrestrial. We've recently sort of published this article here, and this, even though the terrestrial green line here that you can see is to do with islands, it does sort of give an indication of the sort of glaring gap between the progress that's made in pest control for mammals, insects, plants, and what we can do with marine species. There's a pretty substantial sort of gap there that's probably a century old, and there are innovations and sort of jumps in ability and efficacy that happen over time if people are trying to make attempts to remove problem species, and you can sort of see that in the curve that has happened terrestrially. And so we'd like to get the marine curve on an upward bend here, and oftentimes there's it's a difficult environment to work in. There's a perception that it's an open system and that everything is everywhere, and that's not quite true. And it depends on the taxa that are involved in. There are plenty of terrestrial species that have widespread distributions as well, and dispersal abilities. And it feeds into, there's a good literature on sort of the social science behind all of this, that it feeds into sort of defeatism, I think, and uncertainty in action, and essentially not making attempts. And so then the pests that do arrive in places, they proliferate and their impacts compound and continue over time. And so MB have funded a program for us over 5 years to help try to break through some of the bottlenecks that are involved in that. There's sort of three main components to it here that you can see in the three sort of colors. So strategy and tactical component. I'll spend most of the time today talking about eradication technologies and tools. And then there's getting it out into the field and collaborating across broad different types of expertise and abilities to ensure that we have an effect on either the impact that the pest is having or the pest populations themselves, so that we can sort of reduce our the damage that can be caused by these things. In the strategy and tactics component, there's a sort of a review and synthesis aspect to it, to just sort of understand what are the building blocks that we can quickly sort of accelerate through into marine spaces. So that could be literature from and experience from terrestrial as well as previous marine work. There's also a social science component. We've been working with MPI and councils on sort of defining strategies and sort of understanding what our goal setting environment is. And then importantly, there's a sort of a spatial and a temporal tactical sort of component. So if we do have tools, how do we deploy them? How do we ensure that we're optimizing and sort of getting the best bang for the buck when we use them? And then I'll talk about eradication tools. So one of the things in that strategy and tactics component that we're working on is an incursion response simulator. So we're sort of just finished year one. We're sort of over on the right hand side of this diagram at the moment. The details don't matter too much, but there's essentially a modeling component to understand when you have a pest population that's been detected. Is it growing? Is it receding? What are the environmental drivers of that? What are the inherent biological sort of life history versions of that for the pest itself? And then importantly, whether or not a treatment or a tool or something can start also having an impact on the trajectory of the pest population. So there's a sort of population modeling aspect to it. And then over time, we want to start evolving that out into sort of user-friendly app type of environment that we can that people can use broadly to help at least sort of run scenarios against each other computationally, so they can understand where their best investments could be, both in terms of spatially explicit where to target first, or how long to target, or what your kill rate or removal rate and things like that should be in order to achieve success. And so across these technologies and tool development, there's, I suppose, writ large, there's no such thing as a wrong tool, but there definitely can be wrong tools for the certain context that you find yourself in. And the fact there's no, everything's a trade-off. So that's the point about not being a wrong tool, really. So across the bottom here, there's a whole sort of range of different types of tools that are being used or could be used in a marine context. And I'll just sort of have a quick run through various things that are sort of happening in the program or more broadly in New Zealand across that spectrum. But each tool has an efficacy gradient, and some of them have a decent amount of precedence that we can learn from. And some of them are fairly new, and so we don't fully understand, and we have to sort of develop those tools to become effective. There's a specificity gradient in terms of whether you can target a particular organism or whether you're going to have a bit more of a non-target field of effect. And so are you willing to take that shorter term hit and get the potential longer term benefit of removing the pest or the impact? And there's a sophistication gradient and a technological gradient across these different things. Importantly, there's a scalability gradient, which feeds into the cost and the cost and benefit aspects. And then there's a technological readiness level about whether these things would be allowed to be operated out in the wild. Some of them need to have some pretty careful considerations before that happens. So there's no need to spend too long on manual removal. You guys are experts at it. I think it's it's another thing that we might take for granted, where we don't want the footprint of Sabella, for instance, the fan worm, to expand too broadly throughout New Zealand, and not only by managing the vector, but also really jumping on top of any detections that happen in the environment itself, be that the built environment or the natural environment. That's really, really important. And that takes consistent effort and understanding and efficiencies over time, and to just sort of maintain that no new populations sort of sprout up. And I suppose the issue over time with manual tools is that if you miss something and the intensity or the density of the pest becomes too high, then you may overwhelm the ability of manual removal and things to sort of get on top of that. And that's certainly the case in certain parts of New Zealand with Sabella. So if those populations were to be tackled, you'd have to move to a different type of tool. Mechanical tools. So the one example that's pretty obvious here, we've been helping out with the development of the dredge tools that have gone through phase 1, 2, and are currently in phase 3 development up in Northland. When Callirpa is not dying on its own for unknown reasons, this is one way to get rid of 2 hectares of it. That sort of picture shows, but there's a whole lot of science and engineering, and I think one of the key things that's sort of happened because of Callirpa in recent years in New Zealand is this this expansive collaboration across different types of disciplines. You know, having the engineers, having the hydrodynamics people, having the maritime industry researchers, biologists, all working together. And you can really start to understand, you know, what it is about the different components of an overall system to be able to efficiently get your pest out there. And in many cases, you're trying to define a way to have a one-pass system, so you don't want to have to go through the same space multiple times and sort of incur all that cost. The next one is sort of broad spectrum chemicals. Chlorine is the obvious example for this one. We've had a couple of projects that we've been working on. One for MPI is the static version in the sort of 3-dimensional environment like marinas, so the built environment where a lot of our first detections happen. Sometimes you can be curtailed by visibility or other sort of difficulties in terms of being able to hand remove everything. So it's good if you can have a sort of a contained enclosed space, have a short-term pulse of toxicity, and then not have downstream sort of effects of that toxicity. So the chlorine lifespan and the byproducts from chlorine are all relatively short-lived. And so you can have an intense burst if you can sort of have this contained treatment. So we've been working up in Northland marinas to sort of understand what are the parameters you need to achieve in order to be able to kill, in this case, Sabella, and then what would be the scale up costs and sort of versions of that. And also for chlorine, we've been working with Brooke and Sarah McRae and their team on the mobile chlorine chamber that can move along the seafloor in the Callirpa context. And so we've been making some good headway recently, and there's field work coming up for further testing of that treatment delivery. But that's really, those are pretty promising avenues ahead. Bringing Mataranga into the picture as well. In the program, we're sort of working with Patuhara Keke, who are up on the south side of Whangarei Harbour, to look at how pests interact with their tonga species, and how is it that their own sort of suppression efforts and removals can sort of maybe be enhanced, or what the effect of that might be, and how you manage sort of re-immigration from a broader harbour that they don't have the resources to sort of manage as well. So we're working with them through the program, and we'll sort of make some progress on that suppression style angle for an area that already has quite a lot of Sabella, among other things. There's also biogenic control, or a biocontrol, biological control. This has had a mixed, there's been multiple examples of that in New Zealand and around the world, and it has a sort of a mixed history. The terrestrial versions of this are, sometimes there's a tighter bond between the pest and the grazer and the grazed organism. So whether you find that in marine systems is another question. But we do have a long history with Undaria here, and we have a lot of longer term sort of ecological insights now that we can start to take advantage of. So part of the program, we're working, we already know that native species can suppress Undaria, and that Undaria does not actually have the same effect on native species. So there's a timing and a sort of a propagule pressure arms race that you can play there to win. And so we've got a seaweed hatchery work going on to sort of facilitate natives and get them attached to substrates that could then be deployed in the field at scale. And so you can give the natives a bit of a boost and overcome some of the sort of seed bank and persistence issues that Undaria have. Moving along the scale, so we also have mobile species are their own category of issue. And we're working on an AI-enabled trapping tool for Charybdis, the Asian paddle crab. And there's an analogous version of this and that Predator Free 2050 use, where there's a sort of an AI camera that is baited, and the trap itself can sort of identify whether it's a pest or a native bird or whatever, and give it a knock on the head and kill it. So this is the marine version of that for crabs. So we've been developing a camera system. We've been exploring, this is with the Victoria University in Wellington. There's training AI algorithms, and actually the cost of some of the computational components has really come down a lot. So there's a lot of benefit that you can sort of tap into now pretty quickly. And then we've been sort of, some components are sort of 3D printed, and other components we are putting together. So it's about putting these three elements together so that we may end up with a sort of a self-baiting trap, essentially, that can identify and collects data, and then it also can shift the pest to one side and humanely kill it and push the other natives out. And so you sort of end up with a smart trap in some ways. And some of the sort of preliminary work that goes into that is basically training up that AI in all sorts of different light and sediment conditions and all the rest of it to understand where your accuracy level and all of that is going. Aquachemicals. So one of the things, so if we compare aquachemicals to broad spectrum chemicals like chlorine that I mentioned a little bit earlier, the big difference between those types of things would be specificity. So again, the details on this one don't matter too much, but chlorine is a broad spectrum thing that attacks at a cellular level, and there's oxidation that happens. And so that's why chlorine is sort of, well, you can treat your area. And if you're dealing with a monoculture, then you don't have a ton of non-target effects potentially. But it would be great to have something with a bit more specificity. And Patrick just described one of the versions of that in the hull fouling world that targets just barnacles. So there are certain aspects of the biology of certain taxa that you can potentially target. We've had there's herbicides and terrestrial analogs to this that we would like to start speeding up and getting the marine world involved in it. And so in the program, one of the case studies we have is shipworms, which are bivalves. Actually, they're not worms, but they're just a really interesting, quirky sort of species. They've been moving around the world and become invasive before biologists were even recording whether they're native or so. We don't sometimes have a clue really where they originated from because they're so pseudo-cosmopolitan, they call it. But they can have quite an impact, especially on infrastructure, maritime infrastructure and things like that, marina pilings and all the rest of it. And so there's an ivermectin style chemistry that Patrick and the team are working on to sort of get a bit more specificity into our marine pest management. And that may either just target the impact or it could target the populations. It sort of depends on it depends on how stable the chemistry is and whether what the treatment deployment looks like. And then lastly, across this sort of whole range of different types of tools is the more potentially futuristic ones. So gene drives are a potentially, I hate using the term, but sort of a silver bullet, I suppose, that you could rapidly increase the frequency of a genetic element in a population, and you can make that to be deleterious to the pest population, so you could potentially get rid of it. There's been, so nothing's been released in the wild as yet in this world, but nearly everything done to date has been in terrestrial world, and it's often to do with mosquitoes and wasps. The sort of efficiency that happens here is that you can sort of create a genetic modification in an organism and release those organisms into the population. And the top graph there just shows the sort of inheritance of that new gene configuration. So after about eight or 10 generations in this experiment that happened completely in containment, you end up with every mosquito in that population has this sort of gene. And then on the bottom side, you can sort of see the effect of that then is that the amount of eggs, the egg output from the population just crashes down to zero essentially, and you can eradicate your pest. Because nothing's been, there's a sort of a scaffold there to be sort of quickly built on, but still there's certain peculiarities to working on marine species that would have to be sort of worked out, but we're working out on that at the moment. Obviously, this is something that's all happening only in containment. This is not something that's going to be out in the wild. There's been, hundreds of millions spent on the terrestrial versions there, the mosquito and things like that, and those have not yet to be released in the wild as well. So this is a process of essentially understanding the tool and then understanding the controls on it and getting sort of societal understanding and potential buy-in about whether or not it would be released. But the key benefits are it's scalable, simplifies access to the population, it's species specific, and it's a sort of a humane way to kill things. So there's not a huge amount of animal ethics issues. But the challenges are, it can jump. So if something got onto a ship, or it could jump out way beyond the sort of intended range that you intended to put it on. And there are some genetic controls that you can put on that. So there are some technologies that stop, they're called daisy chains, that stop the gene drive from after a certain number of generations. So that technology would have to be developed as well. One of the key things is the resistance, both in terms of our own ability to use this type of technology, but also nature itself does a good job of overcoming genetic changes, especially deleterious ones. And so evolution itself might overwhelm your ability to manage pests in that case, and then regulatory approval is a big one. But we're working with the EPA at the moment, working through the permitting process to do entirely in containment at our facility in Nelson about whether we could make a functional gene drive in an organism. In this case, it's Siona saviniae, which is in marinas around New Zealand. Has been established for a while, and this is experimental work. So this is not trying to create a lab population that will crash. It's inserting a sort of fluorescence gene into it and just understanding how quickly that would percolate through the population if we just keep that population ticking over a number of generations. So overall, there's good progress to be made, I think, on technology and tool development. I think we've probably lagged too far behind for long enough. And so it's really encouraging to be working in an environment like New Zealand's, where, there's been some major investments made recently on tool development. Those tools will not only be useful for what's happening now or the pest that was of concern at the time. They will have a longevity that's far beyond that. And yeah, this is just a quick sort of highlight that you can go on our website, on the Biosecurity Toolbox website, and look at all the various resources, including the ones that Chris was talking about, and with various links, and also the tools that we pop up there. And if you want to know more about the program, we've just had a paper published as well, and we can share that out with everybody. So, thanks.
Any questions?
Jen?
Great talk. Thank you. If one was going to look at, it's nice to hear that some natives would like to outcompete on dairy.
Yeah.
We've got a massive plague of it, like we have done a few of it, and I'm assuming you'd have to remove bulk of that biomass before you introduce that native species. Like it wouldn't outcompete a massive.
It would be helpful. It would be helpful to knock that back. And then the main thing the native does is prevent the seed bank, which can persist for a good while, from bouncing back up. So the combination of some sort of removal, manual or chemical, followed by a scalable amount of deployment, essentially. So biodegradable substrate deployment that gives the natives that sort of head start, essentially.
Yeah.
And then the over sort of 10-year data sets show that if you can establish that initial kernel, that the natives maintain their dominance, essentially, and so on dairy sort of falls back. So that's it. That's the challenge.
How would you choose which natives to introduce somewhere like?
Yeah, a lot of it depends. So you want your natives from the local area, but so it's a case of taking some of those plants from that area, bringing them back to the lab to understand the hatchery sort of mechanisms of it, and then potentially you can scale up in the lab for that, and then bring that down. So you're not introducing even new genetic elements to that area. You sort of keep it all biosecure and focus on that. But there's a lot, yeah. I mean, there's been progress made on gravels and ropes and various substrates that you can infuse your natives into, but there's a bit of work to be done there. But, yeah.